Mendelian Genetics Extended. Like I think of extended as extended play in the video. And we watched The Hobbit in extended play. Is that what it's called? Extended play? Okay, whatever. I'm rambling and the puppies want to eat. All right, so we're going to talk about some more related to Mendelian genetics, some more details. All right, so we have this concept called complete dominance. Now remember, dominance is capital letter, so the dominant allele has information and it is expressed. So even if there's only one copy, so we call this, geneticists call this 100% penetrance, and and so complete dominance. So if there's a if there's a capital letter there, that's what you're going to see. You don't have to see two. You don't have to have two. You can only have one present, like we have with these, and all of these have one present, and so you're going to see the dominance. All right, incomplete dominance, on the other hand, is information from both alleles is expressed. So both alleles are going to show. So in this case, we have the red and the white, and we're blending these. Now, this is what they used to think happened. They used to think blending inheritance. So if you took someone tall and someone short, you get someone medium. And we know that that's not true now, but it is in the case of incomplete dominance. Just think of this word. So you have dominance, whatever the dominance. So let's say red is dominant here. Well, you're not getting completely to red. There you go. Now you're going to remember it, aren't you? So we're at pink here. We've crossed two different colors, and we've ended up with the pink. So that's incomplete dominance. So codominance is where both alleles are expressed in the cell. So they're both equally present in terms of the phenotype. So you have black and white coats in animals, AB blood type in humans, and so we're going to see, you're going to see both. So incomplete, you don't see either one, you see something in the middle. Codominance, you're going to see both of them. Could be, white with black spots or something along those lines. All right, then you can also have multiple alleles. So there are multiple, which means more than two, versions of that same basic allele. The glycoprotein hands, the hands of red blood cells, is a classic example. So these hands, the antigens, identify the blood types. So one dominant allele results in the presence of A antigens on the surface of the red blood cell. So we would have A antigens present. Another dominant allele results in the presence of B antigens on the surface of red blood cells. A third is no antigen, and that's a recessive allele. And that condition is referred to as O blood type. So you can have A, B, AB together, or you can have O, where there's no antigens. So since that is recessive, the gene or the stretch of DNA that codes for these proteins has no code for A or B antigen, so neither one of them is made. All right, so let's go back here. So a universal donor is a person that can give blood to anyone. So since the O blood type here, so this one down here, since it doesn't have the A or B antigens, it doesn't cause a reaction in the bodies of people who have AB, or O blood type. So they're the universal donor. They can give, if you are O blood type, you can give blood to anyone. But if I'm A blood type, and so if I received B blood type, I would die. So you have to have the same blood type. Oh, it doesn't matter. They're universal donors. Universal recipients are ones that can receive blood, which makes sense, okay? So donor, you're giving blood. Recipients, you're receiving it. But they can receive blood from anyone. So since the blood cells have both A and B antigens, then there's no reaction if those cells are mixed with A, B, AB together, or O blood type. So if you think about, let's say that we had, I'm standing here with a box of golf tees, and I have two different colors. A would be one color. Let's say A is orange and B is blue, since those are such great colors, orange and blue. But both are still golf tees. They're both golf tees. So O would be like a hand holding no tee. So we're not holding anything. So we would have, I would have orange in one hand, blue in the other, and then a third hand would come around and show nothing, and then I'd have a hand with orange and blue together, and that would be my AB. And of course, you can see that this would be very important when you're donating blood. All right, pedigrees. I enjoy doing pedigrees. It's like a mystery. I love solving mysteries. So a pedigree, what it is this family history of the trait, of that trait occurrence in some kind of chart form. So you're looking at some kind of trait, and how often does it occur? Where does it occur? And by looking at that, we can track it and figure out, is it helpful? Is it not helpful? What's the, well, not that the trait is helpful or not, but it's going to help tell past occurrences of the trait, and then useful in predicting the future occurrences. This is real important if you have some gnarly disorder in your family, or even if you want to look at, what are my ears going to look like, or what's my hair going to look like, or something along those lines. And so the power of the pedigrees is the ability to predict possible genotypes and therefore phenotypes. Remember, genotypes are the letters like big B, little b. That's the genotype. Phenotype is what you're actually seeing. So what's the chance of having blue eyes in our family? All right, so recessive disorders can only occur, well, first, let's look here. So here's our pedigree. So here is dad, here is mom. Typically the males are square. You have the females depicted in round. And then if it's filled in, that means that that has that trait. If it's open like this, it means they do not have that trait. All right. And so then this is dad and mom. And then you come down here, and dad and mom had a girl, a boy, a boy, a boy. And the girl and the boy, two of the children, were affected or had widow's peak, and two of them did not have widow's peak. And then, so the dominant condition would be here on the left, where if you had a recessive condition, notice as you track it, neither one of the parents have it here, but now it shows up in the offspring. Where here, you have parents have it and the offspring has it. So you can look through each generation and see it. Again, here, neither parent has it, but the offspring does. So that's going to show you that it's going to be a recessive condition. All right. So when we look at recessive disorders, those only occur in the homozygous recessive state. That means it has to be lowercase letter, two lowercase letters. It's rare, and because it's harder to get that. So carriers are organisms that are heterozygous in genotype. So they have a 50-50 chance of passing on that allele for the trait. They're not showing it necessarily. They appear normal because they have one dominant allele. So you can't even tell that they have it, but they're carriers for it. The human recessive disorder, so looking at those, cystic fibrosis, also referred to as CF, and this is the most common lethal genetic disease among people of European descent. So typically you're going to find cystic fibrosis among Caucasians. It affects 1 in 2,500 births. Don't tear yourself up with the numbers here. We're just looking at it in terms of reference, but you're not going to be tested as to how many thousands of births this takes place. In Caucasians, it's 1 in 25 people is a carrier for the disorder. So both mom and dad would both have to be carriers, and they would both be carriers and then pass that on to their child. What it does is this disorder creates a faulty chloride ion protein carrier on the cell membrane in the lungs. The fluid builds up in the lung tissues, and ultimately the people drown in their own fluid. It's awful. They're also prone to getting multiple infections in the lung. They have to have treatments all the time, breathing treatments. There's no cure. It's genetic. There's no cure. So they have to get fluid drained from their lungs. There's medicines to reduce the number of times that has to occur, but it's pretty gnarly. Sickle cell. Oh my goodness, what happened here? Let's get that off. Okay, sickle cell anemia, sickle cell disease, most common among African Americans, affects 1 in 400 births, and the 6th amino acid in the hemoglobin molecule is changed in the primary sequence to make the red blood cell. Basically, this you can see how the shape, this is a normal red blood cell, and then they get sickle shape, that was hard for me to say, and they get stuck, and there are all kinds of things that happen as a result. Painful, because this is the oxygen carrier. This red blood cell is carrying the oxygen, so this can't carry as much, and it's not efficient in transporting it, and so so they have all kinds of problems as a result of that. It's very painful when the points of the cell jab into the walls of the blood vessel. There, again, no cure. It's genetic. And the sickle cell trait is people that are carriers. So they don't have sickle cell, but they are carriers. Now, one advantage is at least they have resistance to malaria. And so you're not going to have people with sickle cell that are carriers for sickle cell that can contract malaria. All right. Let's see. Achondroplasia. Now, chondra, this should look very familiar to you. What does that mean? Cartilage. A is without. Look at that. So a dominant disorder only needs for one allele for this disorder to be present or expressed. So if there is a capital letter there, then you're going to see it. And if an individual is homozygous dominant, the disease is much worse. It's often fatal because they're getting like a double dose of it. Human dominant disorders, again, this achondroplasia, this is also referred to as genetic dwarfism. And this happens in 1 in 10,000 births. And most people are homozygous recessive. And so they're a little bit taller for these individuals. Oops. Another one is Huntington's disease. This is 1 in 10,000 births. Again, has a late life onset, usually in the 40, 50-year-old range. So usually after children are born. If people know they have Huntington's disease, they're probably not going to have children, but or maybe, possibly, they might not. But they don't know until later. So the dominant gene has a locus on the tip of autosome number 4. We're going to discuss autosomes versus sex cells and everything. Family history is very important. So this pedigree would be very important in this. It's a slow degenerative disorder that affects the brain. It's almost always fatal. It's very sad. Lastly, I want to talk about genetic counseling and counselors. These are people who run pedigrees. This is what they do professionally. They look at family history. They look at potential outcomes or carriers. If you have someone in your family with some sort of genetic disorder, you're going to go to a genetic counselor. You want to see what are our chances of having this. Now, this is chance. There's no memory with it. So it doesn't mean that that's exactly what's going to happen. And they would perform a karyotype to help with confirming a diagnosis on this. That's all. Yay!